Hello students, I welcome back to you all to my next video lecture on phosphate solubilizers as biofertilizers. In my previous video lectures, we had studied about Rhizobium, Astrobacter, Azospirillum, blue-green algae as biofertilizers. So today, let us study in detail about phosphate solubilizers as biofertilizers. So what are these phosphate solubilizers? Why these phosphate solubilizers are required? We know phosphates are one of an important macronutrient which is required for the growth of the plant. We know phosphate is one of an important ingredient in our energy currency of the cell, especially ATP, DNA, RNA. They have been made up, they have phosphorus as an important ingredient. So without this phosphorus, life would have not been existed. Hence, phosphorus plays a major role in growth and development of any living organism. In the same way, plants also require this phosphorus. But phosphorus or phosphate as such in the soil cannot be absorbed by the plant in insoluble form, or bounded phosphorus or rock phosphates cannot be absorbed by the plant directly. Hence, these rock phosphates has to be converted into simple soluble phosphates called as these can be done by various microorganisms present in the soil. Such organisms are called as phosphate solubilizing microorganism. It can be a bacteria or it can be a fungi, or sometimes even actinomycetes also has an ability of solubilizing these phosphates. Among bacteria, Bacillus, Pseudomonas, Astrobacter, Azospirillum plays a major role in solubilizing these phosphate. Even various fungi, especially Aspergillus and Penicillium, has an ability of solubilizing the insoluble phosphate into the simple soluble phosphates. Hence, such kind of microorganisms are highly essential to improve the fertility of the soil. Hence, we are studying this particular topic in how to isolate these phosphate solubilizing bacteria and how it has to be produced and how it can be applied onto the field in detail. Because of the application of such phosphate solubilizing microorganism, we can save around 30 to 50 kg of superphosphate per hectare. 32, that is superphosphate, is one of the chemical fertilizers. And also the increase use of phosphate solubilizing microorganism also increases the yield up to 200 to 500 kg per hectare. So let us study the significance of these phosphate solubilizers and few examples of these phosphate solubilizers in detail in this particular lecture. Apart from solubilizing the phosphate in the soil, these bacteria or fungi also plays a major role in promoting the growth of the plant. As you could see in this particular diagrammatic representation, you can see this phosphate solubilizing bacteria also play a major role as PGPRs. What are these PGPRs? Plant growth promoting rhizobacteria. Why we are going to call this as plant growth promoters? Because they have an ability of producing various plant growth hormones. They induce or they produce various plant growth hormones which helps in the growth of the plant, and also they have an ability of producing various substances which are very much important in controlling the phytopathogens. The pathogens which infects the plant, they are called as phytopathogens. They have an ability of doing such thing, that is controlling of such pathogens also happen by producing various antibiotics or siderophores or HCN. Apart from that, they also has an ability of producing various hormones, as I was mentioning before. The various hormones, as we know, can classify this growth regulators as promoters, which promotes the growth of the plants, which are like this: gibberellins, cytokinins, indole acetic acids. These are some of the growth promoters. There is one more hormone called as ethylene. It has been placed in transition between growth promoter and retarders. Retarders are such kind of hormones which retards the growth of the plant. There is one more hormone called as abscisic acid. This is a growth retarding hormone. So this is in general about the hormones. So here these bacteria has an ability of producing this growth promoters, especially indole acetic acid, which is commonly called as auxins, where they play a major role in helping the growth of the plant. They play a major role in apical dominance, helps in the increasing in the length of a root as well as the length of the shoot, and also they play a major role in root proliferation and also enhances the uptake of the nutrition. Cytokinins, they play a major role in growth of the plant, which mainly helps in cell division. Gibberellins produced by such growth promoters, they also help in increasing the length of the plant. These are some of the hormones which play a major role as growth promoters. One more enzyme this growth promoters PGPRs has an ability of producing, that's called as ACC deaminase. This ACC deaminase is one of an enzyme which actually hydrolyze the precursor of ethylene. Ethylene, you know, it's one of a hormone which actually which is being produced by the plants during various biotic and abiotic stress. Ethylene is one of the hormone which is in the form of gaseous form, which is being produced during biotic and or abiotic stress. Biotic stress, which includes when there is a pest attack on the plant, or when there is a drought, flood, salinity, heat, cold, heavy metal in the soil, nutrition deficiency in the soil. During such condition, plants has an ability of producing ethylene. Such time the ethylene production happen. So this what this PGPRs does, they stop the production of this ethylene. How they stop? By hydrolyzing by hydrolyzing a precursor for the production of ethylene, that is aminocyclopropane 1 carboxylic acid. This is a precursor for the production of ethylene. If this precursor itself has been hydrolyzed by production of an enzyme called as ACC deaminase, because of the production of this ACC deaminase by the PGPR, the precursor for ethylene has been degraded. Hence, the ethylene production stop. If the ethylene production stop, the major role of this ethylene is aging of the plant increases. Aging happen due to the production of ethylene. Ripening of the fruits happen. Opening of the flowers happen due to the production of ethylene. Abscission of the leaves. Abscission, that is the Falling of the leaves happen due to the production of this ethylene. Senescence of the plant happen. Retardation of the growth of the plant happens due to the production of this ethylene. If the ethylene production has been stopped by production of this enzyme ACC deaminase by such kind of bacteria, the chances of promoting of age or abscission of the leaf and even the senescence everything can be control. Hence, this phosphate solubilizing bacteria, which also promotes the growth of the plant by producing such kind of growth regulators, and also by producing various substances which can control the biotic stress or abiotic stress. And apart from this, these phosphate solubilizers also has an ability of producing various organic acid. Because of the production of organic acid, the insoluble phosphorus is being converted into soluble form. Such soluble phosphates can be utilized by the or absorbed by the plants for their growth and development. And also there are certain phosphate solubilizers which also improves the metal mobility, especially the zinc metal solubilization, especially the zinc. Zinc is also one of the trace element which is required for the growth of the plant. There are these organisms, by producing organic acids, they also solubilizes the metals so that the roots can uptake this and improves the growth of the plant. This is the mechanism of growth promotion by phosphate solubilizing bacteria. Now let us study some of the phosphate solubilizing fungi which has an ability of producing organic acids. We all know the phosphate can be solubilized by the production of various organic acids. Some of the examples have been enlisted in this particular chart, and also these are some of the workers. I have given the references of them where they have reported various acids produced by these fungi which plays a major role in solubilizing the phosphorus or phosphate. You can go through this, especially the various organic acid produced by aspergillus species and penicillium species. Many species of aspergillus and many species of penicillium has an ability of producing various organic acid which has been reported by various workers. Some of the dominant organisms or the genus includes aspergillus and penicillium, and some of the predominant acids are gluconic acid, malic acid, oxalic acid, citric acid, succinic acid, gluconic acid, tartaric acids. These are the different acids which are involved in conversion of the insoluble phosphates into the soluble form. And these are some of the examples, or these are some of the phosphate solubilizing bacteria which play a major role in producing or in helping the growth of the plant. That is, they has an ability, apart from solubilizing the phosphorus, they also has an ability of producing various growth promoters, and these has been reported by various workers as enlisted in this particular chart. The dominant group of bacteria which play a major role in production of such growth promoters are Bacillus species, Pseudomonas species, as well as Serratia species, Azotobacter species, Azospirillum species, Acinetobacter species. These are some of the dominant group of bacteria which play a major role apart from solubilizing the phosphorus, apart from fixing the atmosphere, and also there are some organisms like that of Azotobacter, Rhizobium. They have an ability of fixing the atmospheric nitrogen, also they have an ability of solubilizing the phosphorus, and also they have an ability of producing various uh, antibacterial or substance or antimicrobial substance to control the pathogens, and also they have an ability of producing various growth promoters where they have an ability of producing various siderophores or ACC deaminase, indole acid, gibberellins, cytokinins, antifungal substance, HCN, metal solubilization, etc. So these are some of the examples of the bacteria where apart from phosphate solubilization, they also play a major role in controlling the pest as well as promoting the growth of the plant as reported by various workers in this particular field. And these are some of the examples of the phosphate solubilizing microbes with biocontrol activity, especially the bacteria Pseudomonas species, Pseudomonas aeruginosa, Pseudomonas putida, Aspergillus niger, Penicillium species, Trichoderma species. They have an ability of controlling the pest by various mode of action, by producing either an antibiotic or a siderophore or any other antifungal substance. They have an ability of producing such biocontrol controlling agent, or various enzyme they can produce. So apart from phosphate solubilization, they also play a major role in controlling the pest. Hence, these organisms can be employed as a biocontrolling agent as well as biofertilizers. So now, how to isolate these phosphate solubilizing microorganisms from the soil? So, like how we are going to isolate the various bacteria or fungi or actinomycetes in the soil, in the same way we can isolate, but the media which we are going to use here is a special media, either a soil extract agar, or a best media which we can use is called as Pickovskias agar. Pickovskias agar is one of a special media, and also it is one of a, we can call it as an indicator media also, which we can isolate the organism using this Pickovskias agar. The composition of this Pickovskias broth I have given here, if we add agar, it's a solid media. One of the important ingredient in this Pickovskias agar is calcium phosphate, which we are going to provide along with all other basal nutrition. Apart from glucose, carbon source we are going to provide ammonium sulfate as a nitrogen source, KCl, magnesium sulfate, manganese sulfate, ferrous sulfate as a trace elements, and also yeast extract for the nitrogen source. We are going to provide this. Important ingredient here is glucose and calcium phosphate. Why we have used this calcium phosphate? So in this calcium phosphate, whatever the ingredient here we have used is an insoluble phosphorus. It's an insoluble phosphorus where the media is quite opaque with the presence of such calcium phosphate. Once you isolate the organism by using rhizosphere or non-rhizosphere soil can be taken as a source of inoculum, and serially dilute the inoculum and plate it on to a soil extract agar or Pickovskias agar. In case of soil extract agar, instead of using calcium phosphate, we are directly using a sterile soil. The soil has got phosphate in them, which is insoluble form, which can be insoluble form. But here we are going to use calcium phosphate to screen them easily. So that's the only difference between soil extract. We are going to prepare a soil extract That is, we are going to boil around 100 grams of soil in around 100 ml of water. Once we boil it, we will filter it. That filtrate we are going to use to prepare the agar. In that filtrate, we can find the phosphate, insoluble phosphate can be found. There we are going to use such soil extract. In this Mikowski's agar, we are going to use calcium phosphate. That's the only difference. Once the soil sample has been serially diluted, it can be plated out onto the media aseptically. Any of the method can be followed, either a spread plate or a pour plate technique can be followed, and incubate at room temperature. Incubate at room temperature, you could see after the period of incubation, you could see the clear zone around the colony. Clear zone around the colony indicates that the insoluble phosphorus has got solubilized. The organism has solubilized the insoluble phosphorus. How they would have solubilized these organisms during their growth and development, due to various physiological activity, various organic acid which was enlisted in my previous slide. These organic acid can be produced by these organisms. Because of these organic acid reacts with the insoluble phosphorus and convert it into a soluble form, because of which we could see a clear zone around the colony indicates that this organism is potential of solubilizing the phosphorus. Hence such strains can be subcultured, purified, identified, and used for the mass production and application onto the field. So this is how the isolation of this phosphate solubilizing microorganism from the soil can be done. The media which we can use is Mikowski's agar. So how to apply this phosphate solubilizing phosphate solubilizers onto the field? One of the well-known method is seed treatment and direct application onto the field. Seed treatment, as we all know, the seeds which we have to sow onto the field, such seed has to be treated with the slurry of the phosphate biofertilizer with water. The phosphate biofertilizers can be diluted with water, and it can also be treated with gum arabica, and the slurry has to be prepared, and the seeds which we are supposed to sow has to be soaked for some time, and later it can be dried in shade, and once it has been dried, it can be used. Such kind of seeds can be sown directly into the main field. That is called as seed treatment method. Whereas in case of field application, 3 kg of the inoculant, that is phosphate solubilizer, biofertilizer, whatever we have brought, 3 kg of which has to be mixed with 25 kg of farmyard manure and 25 kg of soil. Mix these three thoroughly, and such mixture has to be broadcasted throughout the field by top dressing. This is another method of application of this phosphate solubilizers. So the method of application of this biofertilizers in detail you can also see in the documentary, the link which I have provided in this particular slide. Thank you for watching this particular video. Next, I am going to teach you about mycorrhiza as bioinoculant.